everybody. Thanks so much for the invitation. It's really an honor. I would say that I'm a bit of an outsider to the field, so I'm definitely not an expert on NMDA receptors. I come really from the epilepsy field and the kind of network neuroscience aspect of things. So I also changed my talk a bit after the conversations yesterday. I'm going to tell you about our findings with the GRIN2B rat, but also I'm going to tell you about a new biomarker that we're trying to develop, which is also important for the establishment of new therapies to be able to quantify if they're working or not. I want to highlight that the vast majority of this talk is the work of my student, Katerina Ristova. I was joking with Javi last night that basically she's going to leave and my lab's just going to stop because students are really the backbone of what we do. There's a couple of examples of great ones here, David and Sylvia. Katerina has become a brain surgeon, a computer programmer, much better statistician than I am. She's an amazing writer, a communicator, and she won't mind if I tell you she also has epilepsy that is very well controlled. And that was her kind of skin in the game. I think that a lot of us scientists have personal reasons to be involved in trying to understand these diseases. And so even more motivating to be here with you and learning about your families. Thankfully, I don't have to go into this slide very much because we've heard about it a lot. But basically, we're going to be focusing on the GRIN2B rat that was made by the Simons Foundation. David already introduced it very well. I'm quite interested in epilepsy, and there was some disappointment yesterday when I was telling people about this, parents and researchers. So basically, this is a knockout rat, meaning that this gene, one copy of it is not there. It's been genetically modified. And epilepsy is quite common in GRIN2B, but mainly in gain-of-function variants. But as I'll show you, we see epilepsy in this rat. So basically, one copy of it is gone. And my lab, so you've seen as well that us scientists, we're working at different levels of analysis. We've seen from the very first talk from Olivier, people looking at how an individual receptor is moving around the membrane of the cells with these amazing microscopy techniques, but then also these detailed electrophysiological analyses at the cell level, looking what these different versions of these receptors are generating. Then this has an impact on behavior. And David very eloquently told us about the complexity of the brain and how many cells we have. It turns out that it's even more complicated than that because the brain has very distinct areas that do different things and that are organized in very different ways. There's some areas that have very large cells, some areas that have very small cells, some areas where there's more inhibitory cells, so basically the ones that dampen out the activity. And that's really still one of the big frontiers in neuroscience research. How are these different brain regions interacting? And so my lab kind of studies the big output of that. We do maybe the most basic and oldest technique in electrophysiology, which is EEG, the electroencephalogram, which is a critical thing when you're diagnosing epilepsy. Basically, you just put an electrode on the surface of the head. If you close the circuit and ground out electrical noise very well, you will pick up brain waves. And I'll tell you more and more about these as the talk goes on. But this is what we do. So rats, they hate stuff on their bodies, as we saw from the tape test. Unfortunately, David didn't show you videos of it. It's sometimes a bit funny how they kind of try and scratch these things. So basically, there's no way that we could put the equivalent of a human EEG where there's a cap that is put on the head. These, for the animals, unfortunately, we have to implant them permanently. And with what my lab did, the initial years that we began was to screen several of these rat models with different genes associated with neurodevelopmental disorders. And we would basically place what's on that picture on the left, which is essentially a grid of electrodes. The little dots are where the electrodes are located, and essentially the idea is to mimic human EEG, where you have many channels at different locations in the brain. And I'll tell you a bit more later about how these animals sleep and their kind of 24 hours brain signatures. But basically, in order to get a really good picture of what they're doing and how that relates to human sleep, we need to record for 24 hours a day. And to basically optimize that, to allow the animals to move around their cages, we have these fancy EEG machines where essentially they get connected to these implants on the animals, and they send out radio signals to antennae that are outside of the cages. So in GRIN2B knockout rats and in the other animals that we've been studying, we had two basic questions. Do these animals have spontaneous epileptic activity or abnormal EEG? And do they have strange brain state distributions? So basically sleep-wake, is that at all disturbed? So let me show you some videos of the animals. Basically, rats, like mice, they're moving all the time when they're kind of awake. It's very hard to kind of, sometimes with these things that we have on their head, we want to look at them to make sure they're properly implanted, and it's very impossible to kind of pin them down. Unless they're starting to fall asleep. But basically, most of the time, they're moving. So in these videos, you will see they're already in these seizures that we discovered in this rat line, these absent seizures, or petit mal was the ancient term for that. So basically, it's a seizure where the equivalent human seizure is a loss of consciousness and loss of movement, basically. And so this animal is frozen, except for a light shaking of the head that maybe you can make out. And then it'll come out of it. And just like that. And there's this other video where the animal's on top of this cardboard cylinder, and it's in one of these seizures. And actually, I'll end up bashing the side of the cage by accident. And there's a sensory stimulus that kind of causes it to have a reflex, but it doesn't come out of it. See, I hit the head, it kind of jumps and moves. So we saw these things behaviorally. But to prove that they're seizures, you really need to do the EEG. So when they're having one of these absences, when they are in the equivalent non-human state, non-conscious, we see particular signals in the EEG. So what is EEG? It's basically voltages, electrical waves, and usually when the animals are in kind of their healthy, regular, normal brain waves, the amplitude or the size of these signals is kind of around here. This is kind of baseline. So in these electrophysiology papers that we publish, you always want to show what's happening kind of right before the interesting event. And so that's on the left. And then as the seizure starts to emerge, we see these very large spikes, so these kind of peaks. And then here it's too zoomed out, but basically there's like a slight wave. So afterwards, and then another spike and another spike. And so this is called a spike and wave discharge. And you see in human absent seizures an equivalent signature. The channel on the very bottom is actually an electromyogram, which is an electrode that's implanted in the neck muscles. And we use that because to classify sleep, you really need to classify the muscle tone as well, because there's certain sleep states where there's no muscle tone. And you can see that sometimes there's little peaks, which is that kind of small shaking of the head that these animals have. So we have these very characteristic seizures. And what happens compared to the controls? These are litter mates. So basically, when they were bred, you have a heterozygous parent, so one that has one of these copies knocked out of GRIN2B and a wild-type normal parent. They mate, and so half of the offspring will carry the knockout, so we'll not have one copy of GRIN2B. That's the plus-minus sign there. And the other half are the controls. And so basically, we're comparing everything to the controls. And you'll see these plots throughout my talk. So on the x-axis, we have the hours of the day, 24 hours, and we'll have something on the y-axis that we're quantifying. In this case, it's the number of these seizures, numbers of spike and wave discharges. And just so you know, so this black bar on the x-axis is when the lights are off in the animal facility versus when the lights are on over here. And basically, throughout the day, these mutant animals are having lots of these events. So they're bin by hour. Each point is one of the hours of the day. In summary, here, this is what's happening over the 24 hours. And you have a range of seizures across these animals. So each dot is one animal recorded over 24 hours. This was huge N for this kind of study, 18 and 19, which is basically that number of 4-hour surgeries and week-long recordings that Katarina did over the 3 years of work. And essentially, they have many more of these events. So the mutation really pushes this system, the brain, despite what is being seen in many different brain areas that David was talking about, that there's not that many changes that we see in these specific populations at the synaptic level, but somehow at this huge kind of brain level, EEG, we see lots of these seizures coming up. They're also longer in duration. So some of you will wonder, well, why are the wild types having any at all? That's kind of strange, no? And I think that that's one of the issues with animal models. So different species, even different strains or types within that species will have different susceptibility to abnormalities. It might be behavior, it might be questions with memory, it might be seizures. And so rats, and in particular this strain, Long Evans, are particularly susceptible to having spike and wave discharges. Albeit, the wild types have much shorter seizures. They're definitely not as many of them, as over 24 hours. And so the total time that they're spending in over the 24 hours a day is really, really minimal in terms of the controls. But the mutants, over the 24 hours of recordings, they are spending just around 3,700 seconds. So an hour of the day, 3,600 seconds, they're spending in this seizure state. So it's a quite severe phenotype from my point of view. I mean, it's a long time to be seizing. This is another analysis that I want to tell you about that we use a lot in the lab. It's something called a power spectra. And yeah, what is that? What is power? So basically, power is the amplitude or how big these signals are. That's what's on the y-axis. And it's logarithmic, meaning that each jump is 10 times more than the previous jump in data. So basically, it's a huge difference from going from 10 squared to 10 cubed. And on the x-axis, we have frequency. So our electrical signals from the brain are oscillations that are moving up and down, and different brain states have different patterns. They will either be faster or slower, so the frequency of these oscillations will increase or decrease. And so, for example, here, this is during the seizures, both in wild types and in mutants, and essentially, there's this huge peak here at about 7 hertz, which matches, if you look at us electrophysiologists, we're quite minimalist with our graphs, so this is the scale here, this is 1 second. So you have about 7 of those spikes throughout in 1 second of data. And we see that in this big peak of our analysis of this power spectra. But it's very large. It's so large that this algorithm that we use to calculate actually gets confused, and there's a second peak at 14 and a third peak at 21. And these are essentially harmonics. So it's just so large, this seizure signal, that it sees it at multiple frequencies. And we find that if we compare the seizures in the wild types to our GRIN2B hets, that the GRIN2B hets, their amplitude in seizures across all frequencies is much larger, suggesting that there might be different mechanisms in how these seizures are generated. So they seem to have this phenotype of epilepsy. The next thing is sleep. Why is sleep important? Well, obviously, for you, it's important. You see it, there's reports from parents that there are issues in sleep. Sleep, the more and more research that comes out, is basically linked to everything, to memory, to muscle function, to cardiac health. Yeah, you really want to be sleeping well. And it's quite, so despite some differences between rodents and humans and other species, it's very well conserved across the animal kingdom, and especially in mammals. And so there's very clear different aspects of sleep, rapid eye movement sleep and non-REM sleep. And essentially, I've put here pictures of what these different brain states look like. And so by eye, if you look at REM versus non-REM, you can see that there's a difference there. No? This one has very large peaks. It's sometimes called slow wave sleep. And then it has these kind of scribbles afterwards, and then another huge peak. And yeah. REM, it's very constant, very repetitive at a kind of smaller amplitude. And wake, you have a mixture of signals that aren't shown completely here. One huge difference between rodents and human beings is that basically, I mean, they're not, as was alluded to before, they're not tiny humans. No, they are. They have completely different ways of sleeping. In that they basically take naps all the time. I don't know if you have if many of you have pet cats that sleep throughout the day. Rodents are the same, and they are nocturnal, as Xavi just mentioned. So basically, they come out at night trying to avoid predators. That's when they feed, and so basically, so they are more awake during the dark. And so here you can see that. So basically, we've binned or taken the number of minutes that they're spending in these different states throughout the 24 hours. When there's the dark bar, we see an increase in wake. That makes sense for both of the genotypes. We see about the same amount of wake, both during the light and the dark hours. Similarly for non-REM, but for REM, already we can make out that there's a decrease in the minutes that they're spending in REM. REM is really important. It's linked to proper memory function. In humans, it's often described as the dream state. So if people are woken up during REM, they report that they're dreaming, whereas during non-REM, they're more likely to not remember anything. They were just sleeping. And so we can look at the totals here over 24 hours, similar to the seizures, and they have this decrease in REM sleep. And it's mainly due to the fact that the individual REM events or bouts, each time that a REM occurs, that's shorter in duration. And there's some sort of compensation. I've found now getting into the sleep field and in these data that there's always some kind of, if the duration of individual bouts of a particular brain state is decreased, and often you see some sort of change in terms of, if there's a decrease in the number of bouts, there's an increase in duration very frequently. And that's the case actually in non-REM too, even though in total they're spending about the same minutes asleep, the distributions are not the same as in the wild type. So basically, the hets have less number of bouts, but they're increased in duration. And that's also reflected in the wake. So essentially, sleep is not the same as in the wild types. And so it's yet another phenotype that we can try and look at with medication. Another student of mine who really just dedicated herself to very complex analyses, and I'm going to tell you a little bit about that later as well. She managed to do the power spectra. Essentially, with these 24-hour recordings, you get lots of noise, lots of artifacts. It has to be cleaned out and properly sorted, and it takes a lot of skill with programming and data science. And so Melissa managed to do that during her PhD. And here now we have spectra of the different brain states. Remember in REM, there was this little very constant peak, and so that's here. This is 7 hertz, but it's not as large as what you see in the seizures, so there's no harmonics. You just see the peak there. Non-REM, you have very large, it was slow wave sleep, so these huge peaks. So we see much larger low frequency events, but essentially this is the big phenotype that we see, or difference is that across non-REM, basically there's more amplitude in the signal of the hets compared to the wild types, suggesting that there's some sort of amplification or overexcitation during that state. So in summary, we have an overall increase in the number of the spike and wave discharges with an increase in overall amplitude and decreased time spent in REM. So absence seizures. So part of the interest of the lab is to understand the mechanisms of how these mutations are generating these events. So what are the underlying mechanisms? And here's where I want to make a side note just to kind of hammer that point that the brain is composed of many different areas. So this is a cartoon of a rat brain. And basically what we know about how absence seizures works, both in humans and in rodents, come from this type of work in other rodent models. And the main theory about it is that essentially there's some sort of site somewhere in our cortex, the exterior part of the brain, that sends a very large excitatory signal down to the thalamus. This is a central area in the brain. It's almost like a tree trunk. If you imagine the brain being a tree and you have the branches, which are the cortex, this area, the thalamus, it's where you receive the first synapse or the first connection from sensory input. So auditory, visual input, touch, essentially that goes there and then goes up to the cortex. And the cortex, the same, when it wants to signal to the body to do something, to make movements, to focus on something in particular, it will send signals down to the thalamus. In the thalamus, you have excitatory cells that project again back to the cortex. And so essentially you have a kind of a loop of activity there, but it also sends signals down to other subcortical structures, more primitive structures in the brain that were evolved longer ago. Interestingly, there's also signals coming into other parts of the thalamus. This particular region is really interesting. It's the reticular thalamus, and it's full of inhibitory cells. And these guys send something called feed-forward inhibition. So basically a signal comes from here, hits this guy, and says, okay, let's put on the brakes on the activity here. And it can actually shape the output of all of these structures and really control the overall activity. And it's really critical, actually, for establishing sleep. So non-REM emerges from these structures. And there's quite a close link between absence seizures and non-REM. Interestingly, one of the first lines of treatment for absence seizures is a drug called ethosuximide, and this blocks T-type calcium channels. These channels, they open when the cells are really inhibited. So they're kind of like rhythm generators. So essentially a cell becomes active, then it becomes very inhibited, it won't release its neurotransmitter, and then these guys open and drive it back up. And that drug, it's not perfectly clean, but one of the main mechanisms that it hits is blocking these signals, and it's quite effective at blocking absence seizures in patients. So what we're trying to do in the lab is to figure out whether this is the same in our animal model. So what we can do in animals is place electrodes deep inside the brain. Here's a kind of cartoon of how we did that, where we targeted cortical areas, thalamic areas, and areas further down in the brain. And the question is it true? Is the cortex really leading this kind of activity? And we found that in 60% of these absence seizures, indeed, the cortex seemed to be triggering the events. So really, what we think is that it's a good area to potentially target, to potentially look at in greater detail which parts of the cortex might be more affected by these mutations. So this is a quite busy slide, but the take home here is that I'm going to show you now a series of experiments where we're giving different drugs to try and change these seizures or try and block them. One is ethosuximide, this drug that can block absence seizures, and another is memantine, this non-specific NMDA receptor antagonist. Many of you in the audience will ask me, why are you testing this drug? So it's been used as an adjunctive therapy for epilepsy and different GRIN disorders. And so it's a non-competitive antagonist of NMDA receptors. So we thought if there's excess activity somewhere in the circuit, it might be a good one to try. Essentially, all the animals receive these different treatments plus saline, which is the control, and they do it in different orders so that there's not some sort of effect from giving the therapies in a particular way. So we gave this systemically, but also into these structures that we think are generating these events. And so here is the number of spike and wave discharges. On the left, you have the controls. On the right, you have the heterozygous animals. And we find that in both populations, the ethosuximide, which is in blue, is very effective at decreasing the number of seizures, at blocking them. Memantine doesn't seem to do much. But if you look at the duration of the event, so how long these spike and wave discharges are, there memantine does seem to have an effect. So both ethosuximide and memantine decrease the duration of these events in these animals. And so there David was quite instrumental in the interpretation, because essentially memantine is a drug that is activity dependent, so the NMDA receptors have to keep opening for it to work. And so the suggestion is that over these long seizure events, it might be acting over time and therefore decreasing the duration, whereas it won't have an effect on how many times seizures occur. When we did injections, and so this is kind of old school neuroscience, can we change the activity of a particular brain region and what happens? So here now we're injecting into the thalamus, and we find that the thalamus injection is quite effective. Both memantine and ethosuximide seem to block the seizures or decrease the numbers, whereas injection into the cortex really doesn't do much. So here you can see that. Cortex is a very large area. We're not sure if this completely worked or we should be targeting other brain regions. But essentially, we want to try and break down these brain circuits that are generating seizures. So in conclusion, we have these absent seizures, which are not necessarily exactly the type of seizure that patients see. There are some reports of them in GRIN 2B. They have abnormal sleep, and cortex seems to be driving the seizures, and we can see some effect in terms of memantine. I don't know if this really matters too much for the patients. I think that all of these models can be used really to start to test other therapies, and so that's really my feeling that it is a good model. So the summary slide for the families is that we make or we use these rodent models, but there's also other types, zebrafish, for example. Also larger animals can be made, that where we either put these mutations in or knock out certain genes, so essentially eliminate them. We then search for phenotypes. In the case of my lab is brain-wide types of phenotypes. So we see absent seizures, we see abnormal sleep. We think it's a good animal to study. And it allows not only development of kind of novel therapeutics, but also potential biomarkers. And I'm already, I think, over time, but yes, I won't go through the next bit. So I'll tell you about that later. But I just want to thank the lab, especially Katerina, who was really the instrumental person that did these experiments. Melissa as well with her analysis, and the collaborators at Edinburgh, especially Peter Kind, who's the head of the SIDB, and the rest of the team. Yeah, perfect. Thank you.